Hello and welcome back. So this slide talks about the acetylation, which essentially is going to decrease the amount of histones that DNA will attach to, therefore making it more accessible for transcription. You'll express that gene more if you have acetylation. Methylation, and explains the process there, is a way to reduce the amount of genes that are being expressed, and you can read about that there. There are things called transcription factors, and transcription factors are going to, of course, factor in how often a gene is expressed, because certain genes need to be expressed if you're going to be a skin cell, and other genes need to not be expressed, and then certain genes, if you're going to be a liver cell, need to be expressed. And these transcription factors must be present for the RNA polymerase to attach. You can read about them here. We have enhancers. And we also have proximal control elements, and you can read about them here. And they're going to help turn on or turn off genes. This is another slide, and here's another little biointeractive thing that you can look to and watch. And you can also see how sometimes these transcription factors help the RNA strand or the DNA strand fold back on itself, and which is an interesting thing that we'll get to a little bit later. This right here is talking about enhancers and promoters and factors, so it's the same sort of thing again. Now we're starting to get into the lac operon or the lactate gene and how it's personal, how it's, or the LCT gene, excuse me, same sort of thing. Here, this talks about how related genes can often be found on different chromosomes, and they need to be transcribed together. A big example of how this happens is with sex hormones. Sex hormones can control lots of genes on lots of different chromosomes and make them work at the same time. This right here is after the RNA strand is made, you can see the different exons in black here, and you can splice the gene alternate. You have alternate splicing that makes certain things, and it explains that down here, how certain exons are deleted and introns are all deleted to make either this RNA strand to go make a protein or this one. You can see how that can make different proteins. Same sort of thing here, alternate splicing after it's made, another one, alternate splicing. This slide talks about how an RNA strand can only live for so long based on factors. As long as the RNA strand is out there, it'll keep going to ribosomes and keep making proteins that can express certain genes. So if we want that to stop, we have to have some sort of process that can degrade the RNA. And that's what this slide is talking about, how that RNA strand will eventually go away. And that's the same sort of thing here. Getting the DNA strand or the RNA strand to break up and to denature. This is another video of how we're interfering with RNA strands so that they quit making proteins and things like that. Other factors can affect the expression of the gene post-transcription, like chemical signals and the degrading of the mRNA that affects its lifespan. So we already kind of talked about that. That's more of the same things. Proteins that need to be degraded are tagged with a protein called ubiquitin, and that can denature a protein. So this is post-translation. This is after the RNA strand has already made the protein. There is some gene expression that can happen with denaturing those proteins afterwards. And that's a video that kind of shows that. Translation, remember, is the making of a protein. And this is more post-translation control. And it talks a lot about that. Remember to always read your notes down here. And here's a slide which kind of brings some more things together, talking about the denation of mRNA or that, or past translation to degradation of a protein. Watch these videos to help you do the Radiolab assignment. Good luck. Love you. Praying for you.